Hi, I'm Alexander Steele, a PhD student at the NSF IUCRC Brain Center at the University of Houston, led by Dr. Jose Contreras Vidal. This is research on supraspinal effects of non-invasive spinal cord stimulation and is a collaboration with Dr. Dmitry Sayenko, head of the Neuromodulation Recovery Lab, Dr. Jerome Matson, a postdoctoral fellow from that lab, and Dr. Phil Horner, scientific director of the Center for Neuroregeneration at Houston Methodist Research Hospital. Non-invasive spinal cord stimulation has been shown to provide sufficient excitability of spinal circuits to facilitate upper or lower limb movements and improve outcomes after spinal cord injury. While research is currently being done to determine how stimulation affects the spinal motor pools, little work has looked into the supraspinal effects of stimulation. Understanding these effects are important because neural adaptations to stimulation could occur, which can change optimal treatment parameters. This, for example, may explain why the effectiveness of spinal cord stimulation to treat chronic pain has been shown to lessen over time. In this study, we are characterizing the brain's response to non-invasive spinal cord stimulation, or TSS, and functional electrical stimulation, or FES, with neurologically intact participants. To do this, we use high-density EEG to record brain activity under either 15 or 30 hertz stimulation, while 5 different participants performed one of 3 tasks: no movement, tonic movement, or a rhythmic movement task while in the supine position for 30 seconds. This was repeated for a total of 8 trials per task. 3 sessions occurred over the course of 1 week with EEG recordings done at the first and last session. For each participant in the session, we computed equivalent dipoles that matched to the scalp projection of each independent component or IC source. Only ICs where the equivalent dipoles explained greater than 80% of the variance were free of artifactual components and were projected to be inside the brain volume were retained. ICs were then clustered across subjects to find common areas of activation. ICs that were more than 3 standard deviations from the cluster centroid were rejected as outliers. This slide shows 2 of the IC clusters we identified using this method over Brodmann areas 4 and 6, areas associated with motor control and planning. The middle and bottom plots show the average response from each cluster during the 15 Hz TSS tonic movement task in IC space and sensor space, respectively. Stimulus was applied between the spinous processes at the T12 L1 level. Time t equals 0 is when the stimulus occurred, and the neural response from this occurs approximately 20 milliseconds from onset. At 15 Hz, there was 66 milliseconds between pulses, and based on the average, the neural response to the stimulus is still ongoing by the onset of the next stimuli. This may explain why 15 Hz TSS for tonic movements has been shown to provide the largest facilitatory effect thus far. This work shows that TSS causes broad and complex changes in supraspinal networks in neurologically intact participants. There are still several open questions regarding optimal parameters for TSS. This information could help uncover better stimulation parameters based on the brain's responses. Further work will be done to determine functional connectivity changes triggered by TSS and FES. Thank you.